Introducing the GeneSig SARS-CoV-2 Winterplex kit from Primer Design, designed for rapid and discriminatory detection of two SARS-CoV-2 targets, Flu A and B, and RSV by qPCR. An all-in-one testing solution for the winter flu season 2020 and beyond. The GeneSig real-time PCR SARS-CoV-2 Winterplex is a CE-IVD real-time reverse transcriptase PCR (RT-PCR) multiplex assay intended for use with nasopharyngeal swabs, oropharyngeal swabs, and sputum specimens. This multiplex assay provides rapid screening of individuals suspected of SARS-CoV-2, Flu A, Flu B, and RSV infections and aids the diagnosis of suspected disease in patients. The GeneSig SARS-CoV-2 Winterplex kit is shipped Lyophilised and should be resuspended before use as directed in the IFU. The kit contains 4 gold-topped vials of Lyophilised master mix, plus 4 tubes of master mix resuspension buffer with blue lids, 2 amber primer probe tubes, one containing the SARS-CoV-2 multiplex, amber lid, the other containing the flu RSV multiplex, yellow lid, 2 tubes of Lyophilised internal extraction control template, blue lid, 1 tube of Lyophilised positive control template, red lid, 3 tubes of template preparation buffer used to resuspend the primer probe tubes, positive control template and internal extraction control template, 1 tube of water for use as a no template control. The gold-topped Lyophilised master mix must be resuspended in 525 microliters of master mix resuspension buffer. The genomic regions chosen for the detection of these pathogens were selected carefully to provide strain inclusivity for a comprehensive detection profile as well as exclusively to avoid cross-reactivity with other viruses. Our in-house bioinformatics scientists performed analysis on over 100,000 viral sequences in the development of Winterplex to provide accuracy in your results. The 2 primer probe tubes must each be resuspended in 220 microliters of template preparation buffer. The Winterplex detection profile includes strains in the current flu vaccines for this season. Primer Design built the Winterplex from the ground up. The new master mix included is our proprietary formulation created specifically to facilitate robust testing and maintain clinical sensitivity in multiplex products. All the biologically active reagents within Winterplex come Lyophilised. This allows the test to be shipped at ambient temperatures without the need for dry ice safely around the world. With the Winterplex test, our design work doesn't stop once the product is ready to ship. At Primer Design, we operate a daily ongoing genomics surveillance program. This is conducted in-house by our very own scientists to provide security to users of our COVID-19 tests during the pandemic. The positive control template and internal control template should be resuspended and handled away from extraction and qPCR setup to prevent contamination. Each tube of internal control template must be resuspended in 1,000 microliters of template preparation buffer. The positive control template must be resuspended in 800 microliters of template preparation buffer. The Winterplex is highly sensitive with an analytical sensitivity of less than 1 viral copy per microliter for each pathogen. Once the samples have been extracted, with the internal extraction control template incorporated into the extraction protocol, then reaction setup for the Winterplex is as shown in the IFU. Each sample is tested with the 2 SARS-CoV-2 gene targets in parallel with Flu A and B and RSV. At Primer Design, we operate a daily ongoing genomics surveillance program. This is conducted in-house by our very own scientists to provide security to users of our COVID-19 tests during the pandemic. We monitor sequences of the virus that are available on global databases and check them against our PCR designs. This ensures that our specific targets for the detection of COVID-19 maintain a 100% detection profile across all SARS-CoV-2 sequences. This allows for a SARS-CoV-2 test alongside a Flu A and B and RSV test, giving healthcare providers and clinicians the ability to screen symptomatic patients who present with respiratory symptoms. The Winterplex test kit provides the opportunity to diagnose patients rapidly, accurately, and confidently between SARS-CoV-2, Flu A and B, and RSV using only 2 qPCR wells per sample. Interpretation of results is simplified using the interpretation matrix in section 9.2 of the Winterplex IFU. You can find the IFU on our website, or you can email us at inquiries@primerdesign.co.uk.